Well, hello, AP Bio students. This is Shelton here, and I am going to be talking to you about Chapter 8, Photosynthesis. So what you'll actually see is you double check your you must knows and then your notes and your FRQs is you'll actually see a lot of terms in here that are fairly similar to what we just covered in cellular respiration. You're going to see ATP production. You're going to see NADPH instead of NADH. We're going to talk about a proton gradient. We're going to talk about ATP synthase. What we are talking about in photosynthesis is actually how we can do this anabolic activity that's androgenic in order to now build large organic molecules. So that's what we're looking at. We're in cellular respiration. We looked at the oxidation of glucose in order to make ATP. So that catabolic exergonic activity is what we looked at with cellular respiration. So make sure before you take the test that you know what you must know. Also, there are 4 sections. I'm going to try to talk you through each of the 4 sections as we go through. So that way you can also stop and you can go back, especially if you are in that B group and trying to learn material on your own. So for today, as we talk about photosynthesis, always reminded here of SpongeBob doing photosynthesis. He does not do photosynthesis, but you can watch that video if you need a mental break. But when we address photosynthesis, let's get the presentation up here. Hide that presenter view. What we are talking about is first we need to break down and make sure that we are really clear on what an autotroph is. When we talk about an organism that's an autotroph, you guys have probably heard that term before as being a producer, where we talked about producers versus consumers. If we're talking about an autotroph can take inorganic molecules and with the input of energy, convert them to organic molecules. So plants are what you guys already know. They do photosynthesis. They're technically classified as photoautotrophs because they are using the energy from photons in order to be able to convert these inorganic molecules of carbon dioxide and water into larger organic molecules with oxygen as a waste product. Now, in evolution, we'll also address the fact that oxygen was not always the waste product when we talked about photosynthesis or photoautotrophs, making organic molecules. Now, I also need you guys to make sure that you are understanding and you're clear on the fact that it's not just plants that perform photosynthesis. So do recognize that, yes, that's what we're going to be talking about. We'll talk specifically about plants, but also make sure that you are aware that there are other domains and kingdoms that actually perform photosynthesis, from multicellular algae to some unicellular eukaryotes and your protists, cyanobacteria, purple sulfur bacteria. That one, say that 5 times really fast. Purple sulfur bacteria. Good luck with that. But recognize that it's not just plants that perform photosynthesis or photoautotrophs. Now, I do need you guys to be aware of another term that is called a chemoautotroph. When we talk photoautotroph, they perform photosynthesis in order to create organic molecules. A chemoautotroph, they are using energy from a different source. So here's just some examples of chemoautotrophs. They're still making larger organic molecules from non-organic precursors. However, like here at the hydrogen vents, this particular chemoautotroph, they're using this energy from the hydrogen vents at the bottom of the ocean in order to now create or form larger organic molecules. Because remember, we're doing this, these are anabolic activities that are androgenic. They require an energy source from some place. A chemoautotroph just doesn't use photons. Now, we're going to be talking about photosynthesis in plants is what we're addressing. So we are talking about our photoautotrophs, but we also have to make sure that we are recognizing that where we were in our last chapter is we were talking about cellular respiration from organic molecules and oxygen and how these organic molecules get oxidized in the mitochondria, and then we have our oxidative phosphorylation in order to generate ATP that is used for cellular work. Now, how we got those organic molecules, though, is we got them through photosynthesis in chloroplasts. So we are classified because we cannot make our own organic molecules from inorganic precursors. This is what plants can do. They take in carbon dioxide and water, they perform photosynthesis, but since this is this anabolic androgenic activity, they need an energy input. So the energy is supplied through light or through photons. So this is where we get the photo part of photosynthesis. We are synthesizing organic molecules using the energy from photons in order to do this. So this would be your producer. Then we get classified as consumers because this is all we have is mitochondria. So we have to get our organic molecules from a different source. What I do need you to take from this diagram though is please, please, please understand a plant being an autotroph, they have both chloroplasts and they have mitochondria. They perform photosynthesis and then they store their organic molecules away. And when they do need ATP for cellular work, then they perform cellular respiration in their mitochondria. So keep in mind, I've told you guys this before, we need plants. Plants do not need us. So we are those heterotrophs. Now, when we do talk about photosynthesis, photosynthesis happens in chloroplasts. So we do need to understand some basic structure of our chloroplast, remembering that your chloroplast is a double membrane structure. So that is evidence of that endosymbiont theory. There is this liquidy filling, kind of like cytosol of a cell, but in a chloroplast, it's called the stroma. So we definitely need to know what the stroma is, and that is going to be that kind of liquidy filling. This is where my circular DNA and ribosomes would be. And then this is also where there's a tremendous amount of enzymes for the Calvin cycle. So that's a cycle that we're going to learn. Now there's also these membranous structures in here, and these are called thylakoids. And so your thylakoid is this membranous structure that actually is where the light reaction of photosynthesis occurs. This is where the color of your plants is going to mostly be. The pigment of your plants is in these membranes that we're actually seeing here. Now, an example of a pigment that we would find in the thylakoid membrane would be a chlorophyll. So you've probably heard of chlorophylls. There's multiple chlorophylls. There's also other pigment molecules that are absorbing different frequencies of light or different colors of light. Now, we also need to be aware of just basic leaf structure. When we do look at the leaf of a plant, this essentially is the organ for photosynthesis. It's flat, it's broad, it actually has very wide surface area, so lots of light can actually penetrate into these cells that contain chloroplasts. Now, there's this outer waxy cuticle on the top and also on the bottom, but the bottom of a leaf is going to have these little holes that are actually formed by two cells called guard cells that are called stomata. These are really important structures for you guys to know. Essentially, these are the mouths of a plant. And those mouths with two lips, one guard cell on top, one on the bottom, or one side by side, they will open up typically during the daytime. And what they allow then when they're open during the daytime is they allow for the diffusion of carbon dioxide in and oxygen out. Now, also what ends up coming out of the stomata is water, and we'll talk about that, where plants that are in very dry, arid environments have to actually adjust the way they do photosynthesis in order to prevent excess water loss, and also a process called photorespiration they have to prevent. So know what your stomata are, but I also need you guys to know that in between the waxy cuticle on the top that are preventing water loss, here are my cells that actually in each of these cells, there's hundreds of chloroplasts inside that. So the chloroplast is an organelle, and there's lots of those organelles in these individual cells that are located in the mesophyll of the leaf. So this mesophyll here, this is that inner leaf, and if you've ever been, bored during a soccer practice when your coach is just yammering at you and you're tearing apart leaves or you're tearing apart blades of grass, you kind of know that there's this also a little gelatinous-y goo that comes out. Well, that is cells, and then also this mesophyll filling, which has some water and things along those lines. And plus then we have vessel elements as well, and we'll talk about that in another chapter of how water gets transported. Waters and sugars get transported through a plant. But this is basic leaf structure. Know your stomata. Know what I'm talking about when I say the mesophyll. And then definitely know that your stomata, these stomata will open and close at different times of the day in order to allow, when they're open, carbon dioxide and oxygen diffusion. Carbon dioxide in, oxygen out is what we're talking about. Here's just a more up-close picture of this leaf structure where each of these are cells. They kind of look like, sour apple jelly beans. maybe you want to snack on those. But with these, here's the cells, but inside, that is where we're now finding the chloroplast. So a lot of students get confused and they start thinking of a chloroplast as a cell, but keep in mind that perspective, that level of organization. This is the organelle, and we're going to have hundreds of these inside then the cell, and then we're going to have a whole bunch of these cells inside the mesophyll in this space that's inside the leaf. And then my stomata is that connection to allow carbon dioxide, oxygen exchange, and water exchange with the outside world. Now, overall reaction that we do need to be aware of is we need to be aware of our overall reactants and products from photosynthesis. So carbon dioxide is a reactant, water is a reactant. My products then are glucose and oxygen. If you actually think about this, this looks like it's just simply the reverse of cellular respiration, which in a simplified chemical equation, it looks like that, but it is not the same biochemical process. We don't just go from oxidative phosphorylation and now go backwards through citric acid cycle and pyruvate oxidation and glycolysis in order to get this. This is not how it happens at all. Completely different processes, but essentially the use of these molecules is kind of reversed, where carbon dioxide is going to be used to build the organic molecule of glucose, which, remember, carbon and oxygen were the waste products as we oxidize glucose in cellular respiration. These are actually going to be reduced in order to reform an organic molecule. Water is actually going to get oxidized. And so what's going to happen is water is going to break apart, releasing its electrons and protons to become oxygen as a gas, which is also kind of the reverse of what happened in cellular respiration. Remember, oxygen got reduced in order to form water in cellular respiration. So we have relationships between the reactants and the products that are fairly similar, but again, don't get confused thinking it's just a reverse biochemical pathway that happens. Now, remember that in order to build these larger organic molecules, I need an energy input. And in photosynthesis, that energy input is coming from light. Now, here we're also just seeing reactants and products and where these molecules end up going in relationship to my reactants and products. I actually do get some additional water produced, and actually my reactants are 12 waters, but you guys don't need to worry about that. You just need to know my overall reactants. I'm going to start with 6 waters, just like I produced 6 waters if I talked about cellular respiration. So here's a diagram you absolutely positively will need to be able to explain before you take a test. Now, overview of photosynthesis. Now remember, with this diagram, we are inside the chloroplast. What this is showing me here is it's showing me stacks of thylakoids that are called granum. You don't need to know that term, just know that we're looking at the thylakoids here inside the chloroplast. And this photosynthesis is actually taking place in 2 different steps. So less steps in cellular respiration. Yay! So it starts with a light reaction, where in the light reaction, this is where light is going to be used to actually generate chemical energy of ATP and NADPH. Now, it was really nice of them to actually have the electron transporter for photosynthesis have a P in it. So when we see NADPH, we know that we are talking about the electron transporter that's involved in photosynthesis. NADH was in cellular respiration. Now, it's not why there's a P there, but it just helps us remember it. But NADPH is chemical energy carrying electrons and protons to the Calvin cycle. Now, the job of the Calvin cycle is to actually now reduce my carbon dioxide into a larger organic molecule, into what we're seeing here is this 1 to 2 to 1 ratio of carbon, hydrogen to oxygen. This is a carbohydrate, and we'll learn a little bit more about that. Now, my Calvin cycle, being a cycle, it has to have molecules that begin the cycle, and then through that I've got an output, and my output is an organic molecule, and this happens out in the stroma of the chloroplast is where this happens. Now, ADP, so ATP and NADPH, these are energy molecules. Those energy molecules were established during the light reaction, but then that chemical energy is used for this process of building up a bigger organic molecule. Now, these energy molecules, well, they can go back to the light reaction and get recharged. So one of the products, or two of the products of the Calvin cycle in energy forms, is NADP plus and ADP, my inorganic phosphate. Those go back to the light reaction, and the purpose of the light reaction now is to recharge these chemical batteries, essentially. And then the charged chemical batteries are ATP and ADPH. Those go to the Calvin cycle, which actually the Calvin cycle then is my sugar making machine. Well, once my energy is gone, I can't make sugar anymore. So now I've got to go back and recharge my batteries back to light reaction. You can also see in this diagram that this, in the light reaction, is where water is used. So from my reactants of photosynthesis, water is directly used in the light reaction. Carbon dioxide is directly used in the Calvin cycle. And also, oxygen is a product of the light reaction. And then my organic molecules of sugar are my product of the Calvin cycle. So this is kind of an overview to help you remember photosynthesis has two stages, light reaction and then the Calvin cycle. Now, before we talk more about photosynthesis, we need to make sure that we understand a little bit more about the nature of sunlight. So we're now actually moving on to 8.2 is what we are going to make sure we're doing. Actually, let me backtrack really fast. When we talk about ATP production here, the ATP that is produced in this process is referred to as photophosphorylation. And so we'll talk about that again when we do talk more specifics about the details of the light reaction. But this process of phosphorylating ADP is using the power, the energy that comes from photons of light in order to now phosphorylate and create ATP. So make sure too that when you're reading test questions, if they talk about oxidative phosphorylation, we're talking cellular respiration. If we're talking photophosphorylation, we are talking about making ATP through the light reaction of photosynthesis. All right, so let's do a little physics here. So physics is fun. Hopefully you guys end up taking physics before you guys graduate and learn a little bit more about light and waves and some of these properties. Here's what we need to make sure that we are understanding just when it comes to photons and visible light. When I talk about photons, a photon is a packet of energy. That is actually what visible light is. It's also what radio waves are, microwaves, infrared light, UV light, X-rays, gamma rays. They're all photons. Now, those photons all travel the same speed too. They all travel at the speed of light. However, you guys know that being bombarded with radio waves like you are right now, because you've got your phones next to you, versus being bombarded with gamma rays, same amount, gonna have a big problem if I get bombarded with gamma rays. These pack a wallop. They've got a ton of energy to them, and that is because how these photons, these are actually referred to as the electromagnetic spectrum. They are arranged based on the wave pattern of those photons. Energy travels in waves, and actually what makes visible light harmless and radio waves harmless to you is the fact that their wavelength is actually longer than something like this on this end of the spectrum, where they're going to have shorter wavelengths or higher energy. So make sure that we're understanding frequency and wavelength relationship to the speed of light. So what we're looking at here, if we look at just a wave, as we go on the electromagnetic spectrum, photons like gamma rays, they actually have very short wavelength. And what a wavelength is the distance from peak to peak. But your frequency is how quickly I can go through one cycle, the complete up, down, back up to my starting point again. So you can kind of understand where if I've got a very short wavelength, I can also have a very high frequency. Those photons are actually vibrating at a very, very fast rate, where these guys are like, yeah, whatever, we'll get there. They're not vibrating as fast. They're kind of taking the longer wavelength, but then lower frequency. So what I do need you guys to make sure that you're understanding when it comes to visible light, and this is what plants use, is the visible spectrum. This is the part that we also use and we detect. Now there's other organisms that actually use UV and they use infrared, or they can detect those as well in photons. We, however, just see visible light that ranges from purple to red, or Roy G. Biv. What I need you to know is that purple, so this violet, is going to be higher energy than red. And that is based on your difference in wavelengths. The longer the wavelength, the lower the energy. The shorter the wavelength, the higher the energy, is what I do need you to understand. Now, I also need you to understand conceptually, and this is not a spot in your notes in order to write this down, but I need you guys to understand why just the nature of light, of like why plants are green. Plants are green because they do not use that wavelength of light. They absorb all of the other colors, but they reflect off the green light. They can't use it. It doesn't power anything. The pigments don't absorb it. Therefore, that's why they look green. So if you decided to have a greenhouse and just shine green light in your greenhouse, you're not going to have a really successful greenhouse. It's going to be green, but there's not going to be any living plants. There's not going to be any photosynthesis going on in there. There might be a little, but not really. So understand that and kind of that nature of pigments. And when we see colors, it's because those are being reflected and not absorbed. In order to create photosynthesis and drive the light reaction, well, photons have to be absorbed. So that's also where we measure how much light and what colors of light are being absorbed using a machine that's called a spectrophotometer. Now, a spectrophotometer is something we would use in a lab. We do not have one in our lab, but you actually are shining light through different colors, different pigments, different structures, whatever, and it's recording then exactly which wavelengths of light were absorbed. Now, this is a skill that you will have to make sure that you know how to do for your test. You are going to have to be able to read an absorption spectra graph that is produced from a spectrophotometer. Here on my x-axis, I have my different wavelengths of light. So here I've got from violet down to red, so that we can see the colors. Now, what I've actually measured here is through this pigment chlorophyll A, this pigment chlorophyll B, this pigment of carotenoids. What this is measuring is how much light is absorbed for each wavelength as we graph through. So what this is showing, so let's actually just take a look here at chlorophyll A. So what this is showing is that this purple range of light, in the 400s, I get a decent amount of that light being absorbed, which tells us that it can drive some photosynthesis. But for chlorophyll A, it actually absorbs a whole lot of this particular wavelength of light. This transition from purple and blue kind of at about 430-ish nanometers of the wavelength. So this is telling us that this would be the preferred. If I wanted to increase growth in this plant that had chlorophyll A, I'd want to make sure that it got plenty of this wavelength of light in order to be able to absorb that light and drive the light reaction. Chlorophyll B is going to be absorbing light within this range, but it's most effective at absorbing this wavelength of light. If I go from the highest peak down here, that's at about at 460, 470-ish is where that would be. Now, you can also see chlorophyll A and chlorophyll B, they absorb some of the orange and red light as well, but they're not absorbing much of the green and the yellow and this blue. Therefore, plants don't use this color, so plants are going to be looking kind of greenish is what they're going to be looking. Now, carotenoids is another pigment, and if we follow what color, what wavelengths of light a carotenoid absorbs, what you're going to be seeing here is it absorbs purple, it absorbs a lot of blue, a little bit of green, and then nothing else. So guess what? Fall comes and we get really pretty colors. We get reds and yellows now, or oranges, that show up in our leaves. Well, that's because chlorophyll A and B is no longer being produced, but we still have carotenoids. So carotenoids are still absorbing the purples to here, but they're no longer absorbing the reds, oranges, and yellows. So now we're seeing orange. So all you would ever have to do, you would never have to memorize which wavelengths do chlorophyll A, B, or carotenoids absorb. Your job would be to read a graph like this if you were given this graph. So this is called an absorption spectra, is what that would be, or the absorption spectrum for each of these pigment molecules. Now here is an action spectrum. This is already telling you the action spectrum, it looks a little bit different. The action spectrum now is what's the rate of photosynthesis. So this is showing based on different wavelengths, this is how much photosynthesis we're getting based on the different wavelengths of light that it is absorbing. So there's still a little bit of green. So green is actually driving a little bit of photosynthesis, but the rate is much higher when we're here in about this 680, 690 wavelength. And it's also very high when we're here about this 420. And that actually kind of overlaps what we're seeing there on how much is absorbed versus which wavelengths are driving more an increased rate of photosynthesis. Now, here's where we get to the cool part. We're going to talk about the light reaction of photosynthesis and get specific about how this light reaction of photosynthesis actually takes place. So first thing that has to happen is I have to have light absorbed by pigments. So if we take a look here at this pigment, so this is a chlorophyll A molecule here. This is a pigment, this is a color. You can see how complex it is. I got magnesium, I've got these rings of carbon, I've got nitrogen, I've got this fatty acid chain, so it can actually be embedded into the membrane. So I've got a hydrophobic region and a hydrophilic. So this is what is going to be absorbing photons. These different pigments is what they're going to be doing. Now, if they're absorbing pigments, remember these are all covalently bonded, so there's lots of electrons that are in here too. Well, what happens as these pigments start absorbing photons, these little packets of energy, it excites electrons. It actually elevates the electrons to a higher energy state. Well, what goes up also must come down, and so sometimes as those electrons then come back down to their natural energy state, sometimes they release light in the form of more photons, sometimes they release heat, or sometimes they create some movement, which is what we're going to address. So be aware of what we're addressing. Now, in a thylakoid, here's where we're going to talk about a thylakoid membrane. You can kind of see that it's this, it kind of looks like this sac, and it's this flattened sac that's a membrane, and then there's this middle space called the thylakoid space in the thylakoid. For the light reaction to occur, the light reaction has two different clusters of proteins and pigments that are called photosystems. So a photosystem, again, it's a cluster. It's an integral protein complex with these pigment molecules embedded into them as well. And those pigment molecules are absorbing light. Well, what the light is going to do is all of these pigment molecules will actually start passing their energy onto one special chlorophyll molecule. So this is at the reaction center, is where all of the pigments are going to join together and say, okay, we'll pool our energy and we're going to pass our energy now to this special chlorophyll A molecule. Now, what's going to happen here is this chlorophyll A molecule, the electrons there are going to get so excited and receive so much energy that actually it's going to force the loss of an electron. An electron is actually going to be shooting out of it, not literally shooting out of it, but it leaves this special chlorophyll A molecule and actually now gets received by another protein that's called a primary electron acceptor. This is kind of like the electron catcher is what it's like. So I have my reaction center. This is in a photosystem is where this is. And so what I'm talking about first is the one that is actually going to be losing the electron. And we'll talk about this more, but this is photosystem 2, which makes tons of sense because it's the very first photosystem. So we're going to title it photosystem 2, where light is going to actually get an electron to leave a chlorophyll and get caught by the primary electron acceptor. This primary electron acceptor is at such a high energy state, it can now actually pass this electron down an electron transport chain, is what's going to happen here. So that's what I'm seeing in this picture. I am seeing photosystem 2 is absorbing light. All that energy is being passed to the reaction center chlorophyll that's going to lose an electron, and that electron is going to be accepted by the primary electron acceptor. Well, now that that electron is at a much higher energy state, that electron is going to move down the energy gradient. So from high energy state down to low energy state, and it's going to pass through another proton pump. So it's going to flow from photosystem 2 to photosystem 1. Again, it's so dumb that photosystem 2 is first and photosystem 1 is second. Never does anybody ever forget this. And we'll talk about why photosystem 1 is named photosystem 1. It actually was discovered first. And then they went, oh, cool, there's another photosystem. Well, we can't rename it. So we'll actually just keep this as photosystem 1, and then we'll go photosystem 2, and everyone's going to remember that photosystem 2 comes first in electron transport of the light reaction of photosynthesis. But what got these electrons moving was all these pigments absorbing energy, passing it on to that special chlorophyll A molecule, and the electron now getting shot up to that primary electron acceptor. And now that electron is going to move from higher energy state to lower energy state, moving through a proton pump. And this proton pump is going to be pumping protons into the thylakoid space, is where you can see the direction that protons are going to go. Now, I lost an electron here. Well, guess what? I got to get my electrons back. Otherwise, the system is going to shut down. So this is the role of water in photosynthesis. The role of water here in photosynthesis is to actually be oxidized and donate the electrons back to this reaction center chlorophyll. Well, as I split water back apart, well, now oxygen is my waste product. Protons are already here in the thylakoid space. This is why I only need to have one proton pump in my electron transport chain, whereas in cellular respiration, I need 3 proton pumps. I had to get those protons into that intermembrane space. Here in photosynthesis, well, most of my protons are already in the thylakoid space. I just need to get some of these protons that are diffusing out of ATP synthase. There it is again, another ATP synthase that's in the thylakoid membrane. I need to get these protons that are diffusing out back in, so that's why I need proton pumps to keep pumping them back into this space. Now, my electrons in electron transport of photosynthesis, remember this is my light reaction. Electrons are going to flow from photosystem 2 to photosystem 1, creating a high proton concentration inside the thylakoid space. Now, photosystem 1's job is to actually recharge that electron, because this electron went from high energy to low energy, but now I have another photosystem here that can actually absorb more energy from light, but actually just pass that back on to the electrons that already exist. So those electrons are going to get re-energized in photosystem 1. So they're now at a high enough energy state so they can get passed to NADPH. And NADPH can be carrying that high energy electron and protons off to the Calvin cycle. Now my high proton concentration here, well, this protons want to move from high concentration to low concentration, just like any other molecule wants to diffuse. But I now have ATP synthase, this turbine that's going to, with a proton motive force, going to be phosphorylating ADP. As protons diffuse through it, it is phosphorylating and creating ATP. So this is what I refer to as photophosphorylation. So make sure you're also recognizing, if I say photophosphorylation, I'm talking about the making of ATP in the light reaction of photosynthesis. If I'm talking about oxidative phosphorylation, I am now talking about the electron transport and chemiosmosis that happens in the mitochondria, is what I'm talking about. So what I'm seeing here with this flow of electrons, these electrons here are starting at photosystem 2, leaves the chlorophyll A molecule, it's moving from photosystem 2 to photosystem 1, and then ultimately ends up in NADPH. This is what I refer to as linear electron flow. So this is my linear electron flow that is occurring in this process. So electrons start here, they end here. Well, I have to make sure I'm replenishing those electrons, so electrons are going to get replenished by breaking water down, releasing oxygen as a waste product. And these electrons will become part of chlorophyll A, then chlorophyll A will say, no thanks, I got enough energy, you can go now. And they will follow this linear path. There's a beginning and there's an end. Now, however, there is times where that flow of electrons is actually cyclic, and we'll talk about that here, where in times where there's not a lot of water and there's not a source of electrons, what can happen is these electrons can actually, instead of going to NADP plus reductase to form NADPH, these electrons can actually just circle back and then power the proton pump. And they come up here and they circle back and they power the proton pump. They don't go back to photosystem 2, but they end up just kind of circling. And that way we don't need an additional replenishment, and we can at least keep the chemiosmosis process going, is what can happen if I have cyclic electron flow, where the electrons simply cycle between photosystem 1 and the proton pump, photosystem 1 and the proton pump, photosystem 1 and the proton pump. Photosystem 1 is just re-energizing that same electron so it can go back and keep the proton pump going. Now, the disadvantage of that is I now don't have NADPH. I'm getting ATP, but without NADPH, I don't have a supply of protons to now build my organic molecule. So, cyclic versus non-cyclic, the picture here that you are seeing, non-cyclic. If it was cyclic, meaning a cycle or circle, that's just going to be at photosystem 1. And that was actually what was discovered first in the laboratory, was photosystem 1 and just cyclic electron flow, which doesn't involve photosystem 2 at all. So that is why, there you go, your fun fact, why photosystem 1 is named photosystem 1. It was discovered first through cyclic electron flow, and then they found non-cyclic electron flow where the electrons start here, where water is split. Electrons move to photosystem 1, where they ultimately reduce NADP plus to NADPH. So that is the whole job of the light reaction. The light reaction is using the energy from light in order to produce these two chemical energy forms, because this is energy in the form of photons. We're transferring that energy to chemical energy of ATP and NADPH that now can get used in the Calvin cycle. Let's just make sure we're kind of reviewing this. There's a couple of different diagrams that your textbook gives you to help you understand this process. You choose the diagram that makes more sense to you, but in this diagram, you are seeing photosystem 2 and photosystem 1. You're seeing light being absorbed by the pigments and passed on to my reaction center. You're seeing the reaction center lose an electron being caught, so to speak, by the primary electron acceptor. That high energy electron is now passed through electron transport chain towards photosystem 1, which is going to power proton pumps that establish a proton gradient that through chemiosmosis creates ATP. Then photosystem number 1 is going to absorb more light, but it's going to re-energize that electron that originally came from photosystem 2. That electron is going to get re-energized and get to a higher energy state, where now it's going to ultimately end up in NADPH is where that's going to be. So here's my water being split at this state because we have to replenish the electrons that we lost. If this chlorophyll loses electrons, it needs more. Otherwise, the whole system shuts down. There's no more light reaction if I don't have a source for electrons. Here's another analogy. So this is a construction worker analogy just to help you understand the role of each photosystem. So photosystem 2's role is to get an initial electron elevated to a higher energy state. And it's the photons from light that's energizing the electron to a higher energy state. And now at a higher energy state, through a series of redox reactions, the electron can pass through electron transport. As it passes through electron transport, it is establishing a proton gradient that the protons will then diffuse through ATP synthase to generate ATP through photophosphorylation. Now, photosystem number 1 is to absorb more light, but now we're taking that same electron and we're trying to get that electron energized enough to a higher energy state so it can now become a part of NADPH. So now that I have NADPH and ATP, this is where these two chemical forms of energy can be used to build sugar. And that's the job of the Calvin cycle. Now, before we talk about that, though, we need to make sure that we are addressing the similarities and the differences between chemiosmosis and electron transport in the mitochondria and that of the chloroplast. So what I do need you guys to recognize that if we're talking about the mitochondria, cellular respiration, electron transport chain is happening in the inner membrane of the mitochondria. If we're talking about electron transport in the chloroplast, we are talking about the thylakoid membrane. In electron transport of both, we are establishing a proton gradient, a high concentration of protons, and in the mitochondria, that's in the intermembrane space, and in photosynthesis in the chloroplast, that's in the thylakoid space. My ATP synthase, My ATP synthase is going to be in the thylakoid membrane for chloroplasts for photosynthesis, and my ATP synthase is also going to be in the inner membrane of the mitochondria. So here is a classic AP question that they will ask you. In a plant cell, what is the location of ATP synthase? And what most students will mistakenly answer is they will only say in the thylakoid membrane. And what you have to make sure that you are remembering, if they're asking you the location of ATP synthase, you have to remember plants do photosynthesis, plants also do cellular respiration. So my ATP synthase is located in the thylakoid membrane and it is located in the inner membrane of the mitochondria. So that's what you have to remember is the and part when they're addressing plants and where electron transport takes place and where ATP synthase is located and from where do protons diffuse in order to generate ATP. So keep that in mind. Okay, so now that we did the light reaction, it's a lot, probably more details in the light reaction than in the Calvin cycle. And making sure you understand that flow of electrons and how light is used, why water is used. And the whole purpose here is to now generate these chemical energy molecules of ATP and ADPH. So we can make sugar out in the stroma. So the Calvin cycle looks like this. So the Calvin cycle, I'm taking in carbon dioxide and I am kicking out an organic molecule. So what I need you guys to recognize is in any Calvin cycle, carbon in has to equal carbon out. Each carbon dioxide brings a single carbon. So in this case, and this is typically what happens in the Calvin cycle, is the first organic molecule that is produced is a molecule that is called G3P. So glyceraldehyde 3-phosphate. You just need to know it as G3P, this 3-carbon organic molecule. It's the precursor to all organic molecules, so all of our macromolecules that we addressed. So if I am making a 3-carbon molecule, I had to have 3 carbons go in the form of carbon dioxide, is what had to happen. Now, all of these steps in here, I don't need you to worry about these steps of how many ATPs got used, how many NADPHs got used, how many ATPs here got used. I don't need you to worry about those details. Now, if they gave you this diagram and asked you questions about that, read the diagram. The diagram is going to tell you that. What I do need you to recognize is, what do I mean by phase 1 of carbon fixation? And what I mean by carbon fixation is I have to take inorganic carbon and get it to stick to an organic molecule. So when you hear fix in science, like if you're fixing bacteria to a slide, you're getting bacteria to stick to the slide. If I'm fixing carbon, I'm trying to get this inorganic carbon to stick to organic carbon. Think of it that way. Now, there's an enzyme, and this is the most abundant enzyme on this planet. The enzyme is called Rubisco. And Rubisco, this enzyme is actually going to be the enzyme that performs this carbon fixation. It's going to take carbon dioxides that diffuse through the stomata and attach it to this molecule RUBP. So that's what I'm seeing here is Rubisco performs this carbon fixation process. So make sure you know what Rubisco is. Make sure you know RUBP is this 5-carbon organic molecule that's there in the cycle first. That Rubisco, so this is the substrate, this is my enzyme, this then is my product. You do not need to know the name of that product. You just need to know that this is carbon fixation. The enzyme is Rubisco. The original substrate then was RUBP. And this is how I'm getting carbon to stick to or carbon fix to an organic molecule. So now I've got that carbon trapped. I am turning that carbon into an organic molecule and turning it into a useful organic molecule. So there's some reductions that happen, meaning I got to get some electrons added to it so I can make it a bigger molecule. So this is where I'm seeing energy, I'm seeing NADPH, so I'm seeing protons that are going in there. This phase 2 is called the reduction phase. And remember, if molecules are being reduced, that means they're gaining electrons. And so this is what's happening through this process is I am making G3P. I had to catch the carbon first, Then I had to have all the adhesives in order to get that to stick together into an organic molecule. I kicked that out in the form of G3P. I had 3 carbons in, 3 carbons out. The remaining molecules, though, have to be regenerated back to this original substrate of RUBP. And so that's what phase 3 is the regeneration phase of getting it back to my starting molecule. So now I've reestablished my carbon dioxide acceptor. So RUBP and carbon dioxide can bind together, being catalyzed by the enzyme Rubisco, and then through magic or input of energy, electrons, protons, a whole bunch of steps in here, I can reduce those carbons now into G3P. I kick one of the G3Ps out, and I go back and I regenerate RUBP. That's my Calvin cycle. And the 3 steps: carbon fixation first, phase 2 is reduction, kick out my G3P product, and then I regenerate RUBP is what I am doing. Now, we talked about the fact that we got these things in our leaves called stomata. So your stomata are these little mouths of the leaves that allow for carbon dioxide in for the Calvin cycle, oxygen out, that's a waste product of the light reaction, but this is also where water leaves the plant through transpiration. Water is going to leave from the mesophyll space out to the atmosphere because this is a much lower out in the atmosphere. It is a much lower water potential out here. So on a hot, dry, arid day, what ends up happening is a lot of water leaves the plant through these stomata, as the stomata were open during the day for carbon dioxide and oxygen exchange to occur. So what has evolved over time is some different ways to prevent that water loss and also to prevent a process called photorespiration. Because what will happen is if I just went, oh, it's a hot day, close those stomata, which actually when it's super hot, stomata do end up closing. They lose water and then they lose their fill. So they end up collapsing and they stop carbon dioxide from entering, but they also then stop oxygen from leaving. Well, unfortunately, Rubisco, if there's oxygen that starts building up inside the plant, Rubisco will actually grab oxygen and attach it to RUBP. And if that happens, if RUBP and oxygen now bind together, because Rubisco did that, well, what's going to end up happening is this organic molecule is going to get oxidized and used for cellular respiration. So it kind of defeats the purpose. Instead of RUBP and these molecules staying in the cycle, if oxygen binds with RUBP, then this becomes a molecule that gets oxidized and used for cellular respiration. So it's kind of the opposite of what we were trying to do in photosynthesis. We don't necessarily want to do that. So that is what photorespiration is. So C3 plants, and that's what these are called, is plants that perform C3 photosynthesis. And why they are called C3 plants for photosynthesis is their initial organic molecule they produce is a 3-carbon molecule. That's what C3 is, a 3-carbon molecule. So what plants have done is they have evolved different mechanisms to prevent that photorespiration and or prevent that water loss. So here are 2 different mechanisms that we need to be aware of that plants that are in very dry, very arid environments have evolved in order to prevent that process from happening. So especially for photorespiration process. So for photorespiration to prevent that from happening, C4 plants actually have separated out their carbon fixation process versus when they're going to now actually do, so the initial carbon fixation versus where they're actually going to be building the organic molecule. They actually have complete spatial separation of these steps. So there's mesophyll cells, but then there's in the middle, there's these cells that are called bundle sheath cells that are in the middle. And so there are specialized cells for carbon fixation. They fix the carbon dioxide into an organic acid. The enzyme that does this, they don't have Rubisco anymore because Rubisco was fired in this environment for doing photorespiration. So what they have brought in is they haven't brought it in, they have evolved the enzyme called PEP carboxylase. And PEP just sounds like a super happy enzyme. So PEP carboxylase is going to be the enzyme that actually grabs the carbon dioxide and fixes it to an initial organic molecule. But this is in the mesophyll cells. And then there's transport of this organic acid into a whole different location in the plant called bundle sheath cells. Because remember, in C3 plants, everything happens just inside of a chloroplast, inside one single cell. So there's no specialization of these processes occurring. In C4 plants, absolutely. Mesophyll cells do the initial carbon fixation, and this initially fixes it into a 4-carbon molecule. That's why it's called C4. And then it transfers that organic acid then into the bundle sheath cells, where now through chemical process, carbon dioxide can now get fixed again, reduced, and then regenerated in the Calvin cycle. Now, the last type of evolutionary adaptation is called CAM, C-A-M. So I can't even pronounce the crassylation acetic metabolism. You don't need to know that. It's CAM photosynthesis. And CAM photosynthesis, they kind of had an easier process for, instead of having like specialized arranged cells that do one thing, move stuff to the next, what CAM plants actually do is they just reverse when their stomata is open and closed. So during the daytime, that is when they actually risk losing too much water. So they close their stomata in the daytime. Where normally what plants are doing is they have their stomata open during the daytime and they close them at night. Well, what CAM plants do is they close their stomata in the morning. They close them in the AM. Close that morning is what they're going to do. So at nighttime, the stomata now is open. Well, there's no light reaction happening, but what they're doing at night is they are fixing the carbon dioxide at night. They are collecting that carbon dioxide at nighttime, and then in the daytime, they close their stomata. And so now in the daytime, when the light reaction can now produce NADPH and ATP, well, now we can start using that carbon dioxide collected at night to start cranking out the sugar, is what's going to happen. So that is their temporal separation of steps, is just nighttime is when they're fixing the carbon dioxide. Daytime now is when they are actually building the sugars, because they close their stomata in the morning, is what ends up happening. So here's your overview. Because you guys are doing a lot of this work at home, this is why I went super slow, tried to go really slow and be more detailed on your notes in order to cover everything. But you can use this, back me up, speed me up, but I have explained to you all of the details necessary for Chapter 8, photosynthesis, making sure you know the reason for the light reaction, the events of the light reaction, explaining electron transport chain, and also making sure you're explaining chemiosmosis and photosynthesis. But then you're also understanding Calvin cycle, and you're understanding those 3 phases of carbon fixation, reduction, and then regeneration of RUBP is what I need you guys to make sure that you are addressing. So with that, know this diagram before you take the test, and I will see you guys in class to answer any questions that you guys may have. But until then, see you guys. Hopefully this helps a lot in breaking things down, and then we'll see you in class for our hybrid model soon. So let's get me out of here. Let's close it. Let's save those changes and get me closed. Here we go. Find it.